everyone, and welcome to today's live broadcast, Tough Targets, Simple Genotyping, New Solutions for CRISPR, Mitochondrial Disease, Neurology, and Pharmacogenetic Research, presented by Dana Pfister Sullivan, Product Manager at Canon BioMedical. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Canon BioMedical. For more information about our sponsor, visit www.canon-biomedical.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. Now, if you have any trouble hearing or seeing this presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. Please join me in welcoming Mrs. Dana Pfister Sullivan. I will now turn the presentation over to her.
Thank you, Christy, for that very nice introduction. Today I'm excited to present on the topic that Christy spoke about, Tough Targets, Simple Genotyping, New Solutions for CRISPR, Mitochondrial Disease, Neurology, and Pharmacogenetics. This is a quick agenda slide of the topics I'll be going over today. Today I'm going to discuss Canon and genetics, selecting a genotyping technology for your tough targets, HRM screening and CRISPR. I'm going to talk about mitochondrial disease and Novallele, neurology targets, specifically APOE and Alzheimer's, pharmacogenetic tests, and finally, I'm going to talk about how you can find your target today. So let's get started with Canon and genetics, as I'm sure not all of you know that Canon has entered into this field. If that's you, you may be thinking, why is Canon at LabRoots? Is this the same Canon who offers cameras and printers? The short answer is yes. However, we are a smaller subsidiary of that larger parent company. The next question you could ask is, well, do you offer cameras for my microscope or some sort of medical imaging? The answer here is actually no, but today I will discuss what we do offer. Before I do that, I would like to start a poll. So this is the first poll today. I'm going to be doing a couple polls throughout this presentation to keep it interactive with you guys. This first one is just to see who out there is familiar with Canon BioMedical. You'll be given three choices. So you have yes, you have I've heard of Canon but not in the biomedical field, and you have no. And I'm going to give you guys all a little bit of time to answer this question for me. So again, the poll should be popping up on your screen. You should see the question, you should see the three answers, and you should be able to answer it accordingly. And I appreciate your time to do that. And so I'm just going to give you guys a couple more seconds. Okay, thank you so much. So for those of you that are unfamiliar with Canon BioMedical, it is a new subsidiary of Canon, but Canon has always been interested in the biomedical field. In fact, sorry, some technical difficulties here. My screen is not changing slides. There we go. In fact, our first Canon president, who's pictured here, was a doctor. He actually developed a new X-ray imaging device for screening tuberculosis in the 1940s in Japan, when the disease was infecting millions of individuals and current diagnostic X-rays were not cutting it. I think his interest in helping fellow men and women comes from Canon's philosophy, Kyosei, which you can see on this slide, which is all people, regardless of race, religion, or culture, harmoniously living and working together into the future. This philosophy often drives Canon when making business decisions, and I think you can agree that it can easily be applied to the biomedical industry and human health. That brings us to now. Canon is choosing to go back to its roots. However, while imaging for diagnostics has fallen slightly behind, there is this shift for a more molecular approach. So in March 2015, Canon USA launched Canon BioMedical, a subsidiary responsible for Canon's life science and molecular diagnostic products. And in September of that same year, we launched our first commercial life science products, our research-use only Novallele genotyping assays, which will be the main topic for today. Before I dive into more details about our assays, I would like to discuss why you would want to choose Novallele for detecting your tough targets. That brings us to our second poll. This poll is a two-part poll. So the first question I'm going to ask you guys is if you genotype human samples currently. And here the poll should have appeared on your screen. So the question is, do you or does your lab currently genotype human samples? And you'll have three options. You'll have yes, no, but planning to do so soon. And by soon, let's say 6 to 8 months. And then the other option is no. And then once you answer this question, like I said, it'll be a two-part poll. So the next question will be, if you answered yes, what technology do you use? So I'm going to give you guys a few seconds to answer this first question. And now you should see a second question appear, and if you answered yes, you'll be able to answer the second question, which is which technologies are used in your laboratory for genotyping. And there's some common technologies here that I'll talk about in the next couple slides, but that is hydrolysis probe PCR, next generation sequencing, Sanger sequencing, a microarray, mass spectrometry, HRM, or perhaps other. Okay, thank you so much for taking the time to answer those polls. So I think it's easy to agree that there are many things we must consider when you find yourself needing to genotype. And there are different situations that may require you to do so. On this section on the right, you will see 3 of these situations where you may need to genotype a sample. For example, you might be pre-screening. So maybe you're selecting a sample to include or exclude from a larger study. The second situation here is screening. So it's basically the opposite. It's a more retrospective approach. A good example of this might be screening gene edits or CRISPR clones for a specific mutation. And I will go into more detail about that later in this presentation. And then lastly, you could be verifying something. Maybe you're verifying something that you've already learned from a different method. So for example, you could have sent out a sample for full genome sequencing, and you want to verify a specific target that came back. And so when choosing a genotyping technology, it's important to know what type of mutation you are searching for. So it could be a single nucleotide polymorphism. You could be looking at DNA insertions or deletions. Maybe you're looking at somatic mutations, or you're doing copy number research or methylation research. And depending on that type of variation or variations you're investigating, you will most likely choose a specific technology. So while those are the needs a researcher might have, I think it's fair to say you have options. For one, you have to consider the experiment. For example, are you trying to discover new mutations and perhaps looking for the unknown, or are you testing for a specific mutation? Sample number and sample amount can also weigh on what sort of technology you want to use. And lastly, you may consider your resources, not just the amount of people or technicians you have and their skill sets available, but also the budget and cost towards your genotyping efforts. For example, do you want them to spend their time on genotyping methodology that requires a robust knowledge of bioinformatics analysis, or would you rather those resources be applied elsewhere? How quickly do you need your answer? And so after a researcher weighs all of these questions, they then decide on a technology to use. Here you can see some of the methods that they can choose from. There's next-generation sequencing, which is a more whole genome approach. There's Sanger sequencing, a classical approach, mass spectrometry, hydrolysis probe PCR, and then there's two PCR-based approaches, like hydrolysis probe PCR, commonly referred to as TaqMan, and high-resolution melting. High-resolution melting is a more targeted approach. It offers a broad selection of targets. It can detect what might be considered tough or difficult targets. It allows you to test one sample at a time, and it can even verify a multi-target discrimination. And what I mean by that is when there are multiple SNPs that are close to one another, high-resolution melting can distinguish those SNPs in one test. The technology is fast. In most cases, it can be run within an hour, but in some cases even less. It can be run on an open platform, and data analysis is scalable and can be very straightforward due to the availability of controls. Because of these advantages that HRM offers, Canon BioMedical used this technology to develop and launch their Novallele genotyping assays. These assays have a very simple 4-step protocol. The first step being DNA extraction, followed by sample preparation, which requires mixing the assay, the sample, or a control with our high-speed Novallele genotyping master mix. Next is to run those reactions on an instrument with fluorescent detection that supports HRM. Most real-time PCR instruments have HRM capability, and I will be providing an example of those later in this presentation. The last step is to analyze the data. Most instruments support HRM analysis, but we also offer Novallele controls that help make the analysis even easier by comparing sample melt curves to those of the controls to call a sample genotype. But what targets are supported by HRM? Many targets are supported by HRM, including single nucleotide polymorphisms, small insertions and deletions, and what I mean by that are those that are less than 6 base pairs or so. But unlike hydrolysis probe PCR, HRM can also detect much larger deletions, so those greater than 200 base pairs, and we even offer some assays in our portfolio that detect over 7.5 kilobases. HRM can also detect mitochondrial DNA targets, which I will discuss in a few slides, and HRM can also support somatic mutation, copy number, and methylation. However, we currently do not offer any products supporting these types of targets. So in the next couple sections, I would like to discuss where you can use Novallele assay in your genotyping research. Specifically in this section, I want to discuss how HRM can be implemented into CRISPR clone generation research. Before I do so, I have another poll for you. Again, this is a 2-part poll, so I'm going to the first part of the poll should show up just about now. And the first question is, are you using CRISPR in your lab today or planning to begin to use it soon? And so your answers can be yes, I'm using CRISPR now. I plan on implementing it soon. And once again, I think with soon, 6 to 8 months. And then the third option is you do not use or you have no plans to implement the use of CRISPR. So I'm going to give you guys a few seconds here to answer this question. And so depending on your answer, if you answered yes or if you're planning on implementing it soon, another question will appear on your screen now, and that is how you are using CRISPR in your research. So your answer could be something like, I'm screening cell models for specific mutations, or maybe you're doing discovery-based research, or perhaps you're doing both. So again, this second poll is now appearing. You should be able to see the 3 options, and I'm going to give you a few seconds to answer those questions. All right, thank you so much. And again, I really appreciate you guys being interactive with me today. So I'm sure we can all agree that CRISPR is a hot topic. As I'm sure you're aware, CRISPR is a newer, simpler technology often used for genotyping, for genome editing, and it does create genetic variants. For background purposes, CRISPR does have 2 main components, one being guide RNA and the other being Cas9, a DNA endonuclease. The guide RNA determines where insertions or deletions may occur and directs Cas9 to bind to that area. Cas9 will then break the double-stranded DNA and repair that DNA in one of 2 ways. The break can be repaired by non-homologous end joining or homology-directed repair. Non-homologous end joining can produce insertions and deletions, seen here, of variable lengths and is much more random, whereas homologous directed repair, seen here, has the ability to be more specific and introduces precise point mutations or insertions by way of a donor template, seen on the right. So these 2 mechanisms for CRISPR repair can create challenges for gene editing. And when creating CRISPR clones, researchers need to screen the clone population to see which ones acquired the mutation or variation of interest. And due to the 2 repair mechanisms, not all clones will have that specific mutation. There are limited downstream screening options for CRISPR clones. Because CRISPR is introducing genetic variation, genotyping techniques are often implemented into the screening process. Current methods include gel-based cleavage kits. However, this can be considered slow, they do lack sensitivity, and they are very hands-on. Another option is to design your own primer assay or a self-primer design, though this does require significant expertise and it can be time-consuming due to the need of optimization. And lastly, you can use Sanger sequencing. However, this does take time. It often requires samples to be sent out, and it is more costly than the other 2 options. But what about HRM? It is useful for detecting genetic variants as well. And as it turns out, scientists have accepted HRM as a simple and effective way to screen for CRISPR-derived mutations. In fact, a group from Harvard recently published a protocol in Cold Spring Harbor's protocol on how to use HRM for detecting indel mutations caused by CRISPR. And in 2017, that same group published a paper using the method. They tested 30 individual cells for the presence of mutations using HRM. They found that HRM was an effective method to identify fully mutant clones. Another group out of University of Alabama published a similar study and found that HRM was highly efficient at detecting CRISPR-derived mutations in zebrafish. They used HRM for rapid and efficient identification of nuclease-derived mutations to help advance cost-effective, high-throughput methodologies for mutation detection. In this chart here, they break down the results from their HRM analysis on 4 different CRISPR-targeted genes, seen here, 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 and here. And they show that HRM was able to detect all cells with a derived mutation 100% of the time. The Novallele genotyping assays use HRM to detect SNPs and indels for clinically relevant variants. Here you can see how the data analysis from these assays easily make clear what variation has been created, which is an advantage that might not be as clear using other methods of variant detection. In this case, the black curve seen here is the homozygous wild type. The red melt curve seen here is the homozygous mutant, and the blue melt curve is the heterozygous variant. Now I would like to move to another example where HRM can be used in detecting tough targets, specifically mutations in the mitochondrial DNA. So why might that be tough? Well, we have to consider the difference between nuclear DNA and mitochondrial. While there are 2 copies of nuclear DNA, there are greater than 1,000 copies of mitochondria in the cell. Mitochondrial DNA is maternally inherited and has a much higher mutation rate, in fact 10 to 17-fold higher than nuclear DNA. And there has been more than 250 pathogenic mitochondrial DNA mutations that have been identified. But why does that matter? Well, as you probably know, mitochondria are the energy factory of our body. When mutations occur, mitochondria can fail to produce enough energy, which can cause disorders or mitochondrial diseases. Symptoms of these diseases can be found on these slides, but they are often neurological problems such as dementia, muscle weakness, mental retardation, learning disabilities, etc. And common detection methods for these targets are next-gen sequencing and Sanger sequencing, but once again, these can be costly and time-consuming. Because of this, we developed HRM-based assays that target specific mutations in the mitochondrial genome that are associated with some of the more common pathogenic mutations. These are MERF, MELAS, and LHON, and their associated diseases can be found in the far right column of the slide seen here. Our published research has found that HRM mitochondrial DNA detection is as sensitive as the common methods, and it can detect heteroplasmy, which I'll discuss in more detail shortly. It is also a viable alternative to the more costly and labor-intensive options. This is one example of the data from a MELAS assay. The MELAS assay detects mutations in the MELAS gene. You can see some information about the target on the left side of the slide, but what I really want to focus on here is the data found on the right. This is an unlabeled probe assay, so there is a tall peak seen here on the far right that represents the full-size amplicon, and there are smaller peaks on this side of the graph that represent the probe amplicon heteroduplex. These smaller curves distinguish the 3 genotypes. And here you can see the wild type is in black, the homozygous mutant is in red, and the heterozygous state is in blue. This is an example of another type of assay, specifically a small amplicon assay. Unlike the assay we saw before, this assay has 1 peak, and that distinguishes the genotype. This assay detects a SNP in the LHON gene, which is associated with Leber's disease, and that can cause vision loss. I mentioned heteroplasmy before, but detecting mitochondrial DNA becomes even more complicated due to the heteroplasmic nature of the mitochondria genome. It is very possible for only some of the mitochondria to have a mutation. In the presence of heteroplasmy, the ratio of wild type to mutant DNA determines the onset of clinical symptoms. Furthermore, it has been shown that a minimum critical portion of mutated mitochondrial DNA is needed before dysfunction becomes apparent. In other words, the more mutant the mitochondrial genome is, the more the phenotype could be affected. Our research has found that the assays we offer can detect mitochondrial DNA mutations between 9 and 13% of mutant DNA, depending on the mutation, and therefore can detect mutations even in a heteroplasmic population where only a portion of the mitochondria contain the mutation. I just showed you how HRM can be used to detect mitochondrial DNA, and now I would like to show you other targets it can detect. In this section, I will discuss some neurology targets we offer. Canon BioMedical offers over 80 targets specific to neuroscience and associated neurological diseases. On this slide, you can see a handful of targets we offer and the specific diseases they associate with. Today, I want to talk specifically about our 2 APOE targets for Alzheimer's. For those of you that don't know, APOE is a gene that encodes for apolipoprotein E, which is responsible for maintaining cholesterol levels. Because of this, it's often associated with diseases of the heart, such as a heart attack, stroke, hypertension, or coronary heart disease. But what it is most associated with is Alzheimer's disease. We offer 2 specific APOE assays that help distinguish APOE alleles. There are 3 common APOE alleles. They are APOE 2, 3, and 4. APOE3 is the most common allele and is found in more than half the population and considered the wild type. The 2 assays we offer detect SNPs in the APOE gene, seen here and here, that cause an amino acid change based on the variation or mutation. So if you look at the chart, if a sample was wild type for both assays that we offer, that sample could be categorized as APOE3. If the sample was mutant for both, it could be categorized as APOE4. It has been shown that the disease risk for Alzheimer's depends on the person's genotype. These disease risks seen on this slide were pulled from the Alzheimer's Drug Discovery Foundation website. Again, the E3 allele is the most common allele and contains the average risk. The APOE4 allele increases an individual's risk for developing late-onset Alzheimer's, and the APOE2 allele is the least common allele in the population. This next slide shows data from blood samples using one of the APOE assays I just spoke about. You can see that it easily differentiates the genotype by the 3 curves. However, on the right, you can also see that by using our Novallele control sets, so in this graph here, that you can compare the sample data seen here and here to the Novallele control curves in blue. This makes it easy for researchers to have and find a positive control in their genotyping HRM experiments, and we offer positive controls like this for most of our genotyping assays. The last example today that I want to focus on is our pharmacogenetic offerings. Canon BioMedical offers over 90 genetic targets specific to pharmacogenetic research. On this slide, there are a few examples of those. You can see the targets, the variant type, and the specific drug interaction. Today, I want to focus on 3 of these in particular. So 2 CYP2C9 targets and 1 VKORC1 target. In this section, I want to discuss a particular study that was recently published earlier this year from University of Florida. In the study, the lab compared 3 genotyping solutions for these 3 specific SNPs associated with warfarin metabolism. The researchers compared our Novallele genotyping assays, TaqMan hydrolysis probe assays, and luciferase-based pyrosequencing by QIAGEN. They looked at both blood and saliva samples and tested these 3 mutations. A mutation in the VKORC1 gene will cause warfarin resistance. A mutation in the CYP2C9*2 reduces metabolism by 30%, and a mutation in the STAR3 CYP2C9 reduces metabolism by 90%. This slide shows the genotyping data for the blood samples. The overall concordance was greater than 99%. I do want to point out that there was one sample that was determined to be homozygous wild type for the VKORC1 gene by both the Novallele assay and the pyrosequencing method. However, that same sample was undetermined by TaqMan. This slide shows the HRM melt curves for those 3 targets with blood samples. You can see the variants are easily distinguishable. Again, the wild type is shown in black, the homozygous mutant is in red, and the heterozygous samples are in blue. This slide looks really familiar to the last slide we saw. However, it shows the concordance of the data in saliva samples. The overall concordance in saliva was 100%. But again, TaqMan failed to determine one of the samples, this time a heterozygous CYP2C9*3 sample. This slide shows the HRM curves for those 3 targets with saliva, and you can see how they're easily distinguishable. The wild type is shown in black, the homozygous mutant once again is in red, and the heterozygous sample is in blue. Overall, the researchers found that HRM analysis is a fast and cost-effective, reliable method to detect a wide range of mutations. And the comparison proved that the Novallele assays are reliable compared to the 2 common options. Before I conclude, I want to quickly show you how you can find your targets of interest today. This slide here breaks down the products that we offer in our Novallele portfolio that I've discussed. I apologize for it being a little bit blurry for you guys. We do offer 4 different things. So we have the Novallele genotyping assays, which I spoke about, the Novallele control sets, the Novallele genotyping master mix, and the 2x Novallele oligo dilution buffer. And I do want to quickly mention that if you have your own primers and probes, you can pair them with our Novallele genotyping master mix seen here and our 2x oligo dilution buffer to implement our high fast speed chemistry that I have spoken about today. The products are shipped at ambient conditions, and the assays in the master mix can be stored at 4 degrees Celsius. The control should be stored at -20, and each assay offers 166 reactions for 10 microliter reactions. In the beginning of the presentation, I showed you a protocol that requires an HRM-capable thermocycler, and Novallele chemistry can be run on any HRM-capable thermocycler. However, we have tested and validated market-leading instruments found on this slide. You will see common instruments, and some may be even found in your lab, such as Bio-Rad's CFX96 and 384, Roche's light cyclers, Thermo Fisher's or ABI Quant Studio, and QIAGEN's RotorGene-Q platform. Again, I have to apologize that this slide is a little blurry, but if you are interested in seeing the type of targets we offered outside of the ones I discussed today in the presentation, or if you want to learn more about the targets that we offer, you can visit our website at www.canon-biomedical.com. Here you can search by discover area or research topic seen on the left. So for example, if you were looking for one of the pharmacogenetic targets, you could click here, or if you were looking for one of the neuroscience targets, you could click here. We offer around 9 different research areas that you can choose an assay from. Or if you go to the assay database, which is seen on the right, you can search for your particular gene of interest. So you can type in your gene here, but we also offer other fields, including a drug interaction dropdown. So for example, you could click this drop-down button here and choose warfarin or maybe a specific cancer therapeutic like tamoxifen. And once you would choose where you're going and looking for your target, it would bring you to a page similar to this. And you can find your catalog number for the assay and the catalog number for the associated control for that assay on this page in this red box here. And then if you continue to scroll down, you can see example data curves for that assay. Again, you can visit us at www.canon-biomedical.com. The products are available in the U.S. direct from Canon BioMedical and via our distributors in Canada, Ireland, and United Kingdom. You can also email us at contact@canon-biomedical.com to discuss your tough targets and how Novallele can help. Before we get to questions, I would like to summarize by saying that I hope now you can see how HRM and Novallele can help detect your toughest targets. HRM detects a variety of targets, and those of which we talked about today are SNPs, indels, rare alleles, and mitochondrial DNA. And Novallele delivers a bench-validated, simple protocol with rapid results that is cost-effective. It offers genotyping controls, so you are not limited to sample number and can test as little as one sample at a time. I urge you to reach out today to see how Novallele can help detect your toughest targets. One last obligatory slide with disclaimers that I just have to share with you guys. So these products, like I said, are research use only. If you want to learn more about the products, you can visit our website seen here. That also contains user guides and product-specific information. And I do want to mention that Novallele is a trademark of Canon BioMedical. With that, I'm happy to answer any questions you may have. Thanks for hanging in there with me today. I sincerely appreciate your time.
Dana, thank you for that informative presentation. As you mentioned, we will now start on the live Q&A portion of the webinar. Now to our audience members, if you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click the send button. We'll answer as many of your questions as we have time for. Okay, let's get started. Dana, our first question is this offered as a service?
That's a really great question, and we actually get that a lot. The products are sold as reagents only to be used with an HRM-compatible instrument that you have in your lab. We do know that often people do reach out to genotyping core labs to get their results, and we do urge you to talk, if you're interested in this technology, to talk to your core labs about this. But right now, these products are just sold as reagents. They are not offered as a service here at Canon BioMedical.
Dana, what if you don't have my target of interest?
So if we don't have your target of interest, or if you go to the website and you search in the discovered areas or the assay database, you can always contact us directly to see how we can help. So again, that contact information that I shared on the past slide, and I think is here too, is contact us at canon-biomedical.com. But we are releasing new assays regularly, and a lot of our assays that we choose to launch actually are coming from suggestions or requests from customers. So please do not hesitate to email that, contact us at canon-biomedical.com to discuss your tough target.
Here's another question. Can you multiplex using these products?
So that's another very good question that we sometimes get. The assays use one dye only, so with unlabeled and with unlabeled primers and probes. So they are not multiplexed, but they are optimized for high-speed reactions, and we have seen that on some instruments, the PCR followed by melt can be completed in close to 20-25 minutes. Similar targets often have the same run conditions, and so they can be run on the same plate. But again, these are not, they cannot be multiplexed, no.
Okay, thank you. Here's a great question, and it's two parts. First part, what is the best sensitivity of HRM? And can I use HRM to detect somatic mutations?
That's another great question. So research has shown that the sensitivity for HRM can be as high as 100%, and it's usually shown to be between 98 to 100%, and most of the literature I've read and the literature that we've published here. HRM can be used to detect somatic mutations. However, the products that we currently offer do not detect somatic mutations.
Very good. Thank you, Dana. I believe that is all the time we have for our question and answer session today. Thank you again for this informative presentation. Do you have any final comments for our audience before we close?
I just want to thank everyone for listening to my presentation today, and I hope that you saw how Novallele could help answer finding and genotyping your toughest targets. Thank you so much, Christy. I really appreciate it.
And thank you, Dana. We really appreciate your time today. I would also like to thank LabRoots and Canon BioMedical for making today's educational webcast possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Goodbye.